Thank you. So just to give a brief introduction about myself, I head the Cancer and Epigenetics Laboratory at the University of Canberra, where I'm also the founder and director of EpiAxis Therapeutics. So our overall goal of the work that we do is really to develop novel therapeutics and diagnostics for really unmet cancers, such as metastatic breast cancer, melanoma, and lung. And I'd like to give you a little bit of an insight of what we do. This is my amazing team that's behind the work that we do. And we work importantly very closely with the oncologists at the ACT Health, as well as the technology companies, which are crucial if we are going to move and advance in this whole area. So what's our focus? Our focus is in particular breast cancer and metastatic breast cancer. Unfortunately, 90% of deaths in breast cancer are due to metastatic breast cancer. And metastatic breast cancer is the spread of the cancer from the primary site to the distal organs of the body. And really, there's about 16,000 metastatic breast cancers are diagnosed just in Australia annually. And this is a worldwide disease. And unfortunately, the survival rate, the 5-year survival rate, is very low. And so what we're really doing is really to address this issue both from the therapeutics angle, but also towards early intervention and diagnostics, which is crucial for patient survival. So to address this gap, several years ago, what my lab and several of the labs worldwide uncovered was that within that primary tumor mass, there's these special tumor cells known as the cancer stem cells. And what we've shown is that these cancer stem cells are crucial and critical for relapse, resistance, and metastatic spread. And so what importantly these cancer stem cells do is that they're resistant to all standard of care chemotherapy. So what they do is they kill the bulk of the, yellow cells, the tumor cells, but what the chemotherapy doesn't do is attack the cancer stem cells. The animation's not working. But what the chemotherapy does is it leaves behind the cancer stem cells, and these cancer stem cells then stay dormant and alive, and whether it's a few weeks or a few years, they reseed and cause the metastatic spread. And what the major breakthroughs of what we've done is we've developed the first-in-class cancer stem cell drug worldwide, which completely kills the cancer stem cells. And so what our novel approach is that we're using the combination of a chemotherapy drug together with the cancer stem cell drug to not only destroy the primary tumor, but also prevent the relapse. And what I'm excited to inform you is that we've now got the green light to go ahead with a clinical trial, which will be the first of its kind worldwide in terms of a combination therapy, and we'll be recruiting our first patients very soon. And we're taking a very similar strategy for other aggressive cancers such as melanoma and lung. In parallel, what I strongly believe in is that we have to have early detection, early diagnostics for patient survival. And to do this, what we've been developing is these very novel platforms, which are really what I consider are a series of blood tests. And what I'm excited to inform you is that we've got one test, for example, that can detect one cancer stem cell in one vial of blood for the very first time. And so what this does importantly is it allows us to track patient progress as we go along and also change the therapy as we go along. And this is just a little snapshot of what the data looks like. So you can see all the lighter colors are the cancer stem cells in the patients, whereas the healthy controls here have no background as well. So this is one of the first tests of its kind worldwide. And so what I would really like to discuss with you, I hope, is that what we really wanted to push forward is work with technology companies to be able to automate these platforms so we can bring it to the patients who deserve it the most. So thank you.